the vessel where it is, or
What's that?
maybe hold the vessel where it is.
Yeah, you can hold it there for a bit. I'm just coming up slow.
Pretty jar OVL, I'll stop you right there, please.
Oh, stop.
Thank you, Tony.
Can we see what this is? Whoa.
Caught on to us.
What's that?
So it caught on and started swimming away.
Can we see what that is? we try to zoom in on that? Oh, wow.
Yeah, I'm trying to keep it outside the water so it's easier.
Straight drifting again, Tony.
Well, basically.
Thanks.
Do you want to try to collect this one?
Sure. Which one we go for?
I guess six.
Six?
Maybe. See it all.
Ready?
I think so.
That's in
Nice.
there.
Oh, wait. Did we get
Oh,
it?
wait,
No,
yeah,
no.
that was in there.
No.
It's right
No?
above
It was,
Oh, man.
because it looked like it. It looked
Sure
like it
did.
from that camera, but then when I looked over here, I
Oh,
could
no.
see it.
Yeah, it's right there.
Where?
Right there, right there, right there.
Oh, yeah.
Oh, it's way up there, okay. Top of the science camera. That's below.
Yeah.
Very tiny. What is that? I think it's
Maybe you can see it.
I don't see it at all. I thought the one on the outside to the right.
Right, so.
Thought. See it. Totally lost it.
We see another one, sorry.
Is that everything?
That's
Is
one.
it? backing you.
Mm-hmm.
Oh, it's Salps.
Right back in that. Look at the,
Wow,
Nice
they're beautiful.
The lighting's good.
Hmm. Oh, look, there's a little animal. There's like a little amphipod stuck to
Mhm.
that
Up on-
that Salp. Yeah, hitching a ride or eating. No, look, there's another one. That's really cool.
You guys know how to do this thing with these all
Wow. The orange parts are their gut. I mostly see their poops in my samples.
No.
Yeah, they're big poopers.
Everybody,
And they sink really fast. Their poops sink like 1,000 meters a day.
Oh, wow.
Yeah.
Wow. They're dense.
Yeah.
it's solid.
That was beautiful.
Yes,
It was so long.
No way.
Wow. What do you think that is? Do you know what that is?
Mm-hmm.
Wow. Cool. I wonder if it's a Foram. Ooh, too close. I would guess Foram. Oh, it's gone. It's all right. right, somebody in the chat asked how deep the deep scattering layer was here, so I texted Anela, who's been monitoring that pretty closely, and she said that at night there's a layer from 750 to 1,000 of very deep non-migrators, and then a layer from 500 to 600 meters, and then another one at 200 meters. And let's see, where are we at now? We're
658.
yeah.
I'm just slowly going up, so
Mhm.
tell me if you want faster or...
Oh, this is fine. Yeah, so it seems like interesting layers might be where we are now. Or I guess, yeah, 500 to 600. So maybe we could go a little shallower, but I mean, we're seeing lots of cool Tinos here. Ooh, larvation. It's still in its house. You see it swimming in there? Ooh, so close.
By the time I see it, it's
Yeah.
almost in the camera.
Yeah.
Science camera, and I looked one of the HDs, and it's like
Impressive. It's very impressive.
Do any of you need a chocolate delivery?
I'm so hungry. I was tempted to go up to the mess and get some nuts, but
Yeah, I saw them refilling those nuts. We go through those pretty quickly. Oh, that looks like the source of the ring we saw earlier. It's like the same thing. See it? Ooh,
Yeah.
what is that? It's the same thing we saw earlier. That's cool.
what if it's eggs?
Yeah, that's what I was thinking. Like, I wonder if it's like an egg, mass, or it doesn't quite look like an organism by itself, right? Does it? It's too fragile.
How barely moving. It's
It doesn't
Yeah.
look like it's
far off,
foreign.
too.
Yeah. Could see. Is that a Salp chain down there? Cool. I wonder if we see Salp fecal pellets in our sediment traps. Maybe.
What's the longest you've ever had a sediment trap out for?
Oh, well, like these drifting ones, I mean, you don't usually want them to be out for more than like 4 or 5 days, just because stuff starts to rot
Oh,
in them, unless you put a lot of fixative. Usually these drifting ones, it's like in Monterey Bay, we only put them out for a day because, yeah, fluxes are so high. But here we're doing it for 4 days because we think the fluxes will be really low. But yeah, with the amount of fixative that we put in them, it's not really good to leave them out for more than 5 days. But then we have ones on the seafloor that we put like a lot more concentrated fixative in, and those they stay out for like a whole year.
Oh, wow.
And they rotate. It's not all collecting into one container. It rotates containers over time. Oh yeah, what's?
Oh, no, he doesn't. That's
No.
just his legs.
Oh wow.
No.
Tell I zoomed in.
Yeah, I
He was
did.
like,
Yeah.
Probably heard it. Felt it with his tentacles. Oh, you have a vessel moving still?
Yeah, I'm gonna move in now.
Alright.
Stopped and just put him into a drift. Hey Tony, I'm gonna move you to that next station, please.
Copy that.
You can go.
Hmm. Ooh, what's that? Is that a Foram? Probably. Oh, wow. Yeah.
You want it?
I think it's okay.
You said you're okay?
Well, is it easy? Would it be? It's collectible? Yeah, let's collect it.
Okay.
This will be the Forum collecting.
You wanna go, Doug?
Oh yeah, look at that.
What's that? Suction samples? Think so?
I don't know how well it would handle the
I think it would be fine.
suction sample.
Yeah, I think so.
I restarted the street.
Although I don't know. We can try it.
Everything else is the same.
Doug, visual.
I think so.
Oh, there's. Oh, yeah, there it is.
See it there.
Yes, you're right.
Jar
Yeah.
6. I'm coming to the right.
Or do you wanna, I just have this right here for you, you wanna slide it? Or
I'm
just
trying.
Go to the right. It's down.
We have to turn the jar light on. Yeah,
No problem,
yeah.
that's right.
Come
Yeah, it's
on.
down.
Yeah, I lost it to the right.
Good shot. That's well bet. I'll get it out the picture though.
Breaking out the vacuum.
Oh, can you look down? What was below? something neat down there. Just a glimpse. Oh, there it is. What is that? Oh, yes. Can we look at, see if that doesn't fly away? I'm wondering if that is this type of Fodarian, which is like one of those deep sea protists, or it could be something else, but ooh. Oh, cool. Yeah, it totally is.
What is it?
Like sometimes we call it the spider rad, but it's like a type of Fodarian. It's
Oh,
a
yes,
yeah.
No, it's like even in and out.
Yeah. Yeah, I totally saw those earlier. There was two stuck together.
Hmm?
I thought I saw it. Yeah.
Which call?
Oh.
They went inside that.
Oh, inside the
Oh.
net.
Yeah.
All right. Well, if we see one of those again, we should try to collect it. It's pretty small though. that a fish?
Mhm.
Looks like, probably.
Whoa.
Ooh, disappeared. Or was that just some kind of reflection?
It definitely disappeared.
Yeah, it probably swam away. Maybe it just swam away. Can we see if that's something? It's probably just a marine snow or something, but I'm just interested by the two things stuck together here.
It's got dangly bits off.
Yeah, can we look at that? I think that's more of the spider rads. Cool. Oh, it's so neat. There's two. Like I saw two of them stuck like that earlier too. That is so cool. Wow. They're really small. Oh. If you want to try to collect them, I think they'll be challenging, but it would be really cool to get those.
Suction or drop?
Either one, whichever you think is simpler.
You want to do that?
Well, right there.
This side. Alright, stick it out here and let you fly.
I see them still. Right there.
Those cameras.
Oh, look apart.
That close. Wow.
I think there was there's just another one because there's two, and then that was a separate. Yeah,
So
there was
that's-
a third one.
That is there, and I'm on 6, so if you get a chance to put the thing, you do that too.
Well, I'm trying.
They're very small.
Of course it gets stuck.
Yeah, there it goes.
Fits in?
What?
No, it's
Oh, on the other side?
no, it's next to the suction sampler. You see it right
See it
above.
there, yeah.
Yeah.
It's in.
Is it?
Yeah, just flew in the jar, but then flew back up.
Did it?
Yep. I just saw it go
Oh,
down the bottom of the
I-
jar, and then go
really?
back up toward the hose.
I thought it saw it go behind
I saw
it.
something fly in there that looked a lot like it.
Huh.
There's something in there.
In the jar?
Yeah.
I mean, something sizable. Definitely we'll find in there. About 15 seconds after. Right there at the hose.
Huh.
I don't know if we video that or not.
It wouldn't come out.
It went down. It might be at the top,
Might be
top
on
of the jar.
on the filter.
Lid.
Oh, you have the bottoms in these. Do you have the bottoms on the jars?
These?
Yeah.
Yeah, they're just talking about.
Oh, you were section sampling it?
Yeah.
Oh, I thought you were. Okay. Yeah, I did see it disappear.
Yeah, and then I
Oh.
something nice size something went flying.
Oh yeah. Well, there's something in there, but I don't know. Yeah.
Well, whatever else I go in there went back up and then come back down.
Yeah. Well, we can see if there's anything in there.
Yeah. See?
Wouldn't it? How long does it take something to get through the hose?
Not very long.
About the time of it.
I see.
I'm gonna go ahead and index that. So
It
it
seems-
is in there.
Yeah.
It seemed a reasonable.
Yeah.
I saw it and then I didn't see it.
Yeah.
That's when you
Yeah.
said there was
I-
something.
That's
saw
me
it.
too.
I saw
Yeah, it suddenly
disappear over there,
disappeared.
So I
and
looked
then-
over there and I saw something
Ah,
fly in there.
Well,
cool. Well, we'll look for it.
Fingers crossed.
Yeah.
It was Wiley.
Mhm.
Caught.
They're tough. Oh, I'm supposed to be logging this. Apology sample.
Possibly something.
What did that go into?
Suction sample jar 1.
Okay. It's cool. We saw quite a few of those at this depth.
No.
Okay, long Long thing or krill? Let's go look after the long thing. Probably a Salp chain. Beautiful.
Let's do this shots up here, and I'll bring you a little tight.
Okay.
Yeah, you can see there's an amphipod in one.
Mhm.
Like you can see there's, I just see it in one, the big amphipod. I guess no, oh, I see a
There's
few.
of them in
Yeah,
there.
they're kind of around the, well,
And there's two in the middle and
yeah,
one to the right.
yeah, I see them. That's cool. Okay, all right.
Bring
I am not supposed to use the word krill. They're decapod shrimps.
Oh.
You can use the word krill, just not for shrimps.
Krill only?
Just for krill.
Scientist.
I'm learning about animals.
Cool.
Ooh, it's probably the, huh. Oh. Oh, it's just the end of, what's at the end of that string? Whoa. Keeps going up. Wow. Ooh. Looks like a ctenophore to me. Wow. Ooh, there's one. There's a lot of them at the steps. Those Fodarians, wow. Keep running into them. I've never seen this many before. I mean, I guess there must be just some locations where they're most abundant. A salp and a suggested shrimp. Wow, so many beautiful salp chains at this depth.
Hmm? Okay.
Oh, look, it's stuck. The spider rad, it's stuck to the camera. It wants to be collected.
Up to the houses.
It really wants to be collected. Please collect me.
Oh, you can just stop. If you
That
stop
was an
there.
arrow. Yeah,
Okay, I'll make
yeah.
my way up there.
Yeah, you've been there for a while.
What do you think that is? Is that some kind of... Huh. What's on that thing? is that?
It's a type of mollusk.
Oh, wow. Oh, cool. Is it like a pteropod?
Related to the heteropod.
It's a sea elephant.
Sea elephant.
Yeah, this one I think is.
The heteropod.
Oh,
So
you
they
two just
swim
snuck in there.
They swim on their backs, so the fin is on the top. And this one has tubercles. You see, it has all these bumps on its back. We've been getting, I think they're Pterotrichus guttata. There are a couple of groups of sea elephants. This one is, it's got like a expanded bib on its back with all these bumps.
Are you speaking into a microphone?
I can hear it.
Kind of.
Oh, okay.
Yeah.
Because people are, yeah, so people can hear you.
Oops, sorry, wrong button.
We have a lot of control.
Wow, look at that middle kind of appendage. It's like flapping around like a figure eight.
That thing in the middle is its fin,
Uh-huh.
and then the vertical, the white thing in the back is its stomach.
Oh,
Wow.
And then, so it has its, like a proboscis trunk that is vertically oriented right now, and at the end of it is a radula with a rasping tongue. So like other mollusks.
Wow. What does it eat?
It's a good question. Other jelly, they're
I don't know.
gelatinivores.
I think it's possible.
Wow. Is it tropical? Or is it in places outside the tropics?
We get them in the California current,
Okay.
but maybe the tropical ones get really big. Or ours are also relatively big.
You want it?
Just gone.
It's gone.
No, we collected a bunch at the mock nest today.
Oh, yeah?
Yeah.
Oh, cool.
Probably the same species.
What were the little green rads that you got, or that you thought you got?
They're in the fridge for you right
Oh.
now.
Oh, are they the spider rads?
No.
No.
I don't think so. Unless.
I don't know, they could lose all
probably
their.
just got torn off.
Yeah. Well, probably it would.
But our spider rat's green.
No, none of them are green. So, I don't know why. They're probably green because they got coated in whatever was in the net.
They're like kind of brown. The core of them is a brownish.
Yeah. Then that's probably a Fodarian because that's their gut, or less like their digested material.
But the Collidarians they're kind of green.
Oh.
Greenish.
Oh, right. Okay. But yeah, but those aren't Fodarians.
What are they? They're still radiolarians?
They're radiolarians, yeah.
Did you already talk about the difference between the two?
Yeah. Well, like a week ago.
Oh.
Cool. Oh, yeah, what's that? Maybe that. It's so. Huh. Is that like what you collected?
A
Oh,
lot-
cool.
a
That's
lot like
I
it.
think that's a Collidarian. Although I usually think of them as being surface because they've got all those dinoflagellates inside.
Yeah, we got them net 2. There were some from 2.
Yeah, we got some a deep handful, and then we got a
Wow.
shallow pile of them.
Huh. I wonder why the deep. I didn't know they were deep.
Contamination.
Maybe they were
No.
sinking.
No, we're at 600
Well, what depth is was net 2 targeting?
If we can collect that one, I would take it.
Yeah, it's gone.
Ah. Snooze, you lose.
Yeah. We're gonna wait around.
Here it is, sir.
But yeah.
Well, how do you want to collect it?
The D sampler.
I have no idea. I don't know where it's at. Oh, it's right in front of the camera.
Oh yeah, it's like really close to the camera.
Go for 6.
I'm just trying to get close to something.
2.
I guess.
Down. Right there it is. Your signs. Six.
Very tiny.
Very tiny.
You see it?
It's behind jars like inside of them. Yeah, that's
No.
Yeah, I don't know where I went to. Come back out. There it is right there. Good job.
You see it?
Do you see it anymore?
I do not.
Yeah, it's okay.
Right there.
Oh, it's out front.
too? Okay.
Manger. Sorry, I lost it.
It's all right.
Pretty close to the bus, I'll get it moving. Pretty drove, you have the waypoint for you?
Copy that.
Maybe.
Get that a little bit close. It's just there.
Yeah, I think it's under the ridge. I know it was just there.
Maybe it was, yeah. Both sides. Nothing crazy.
Hmm.
Dang it.
Two more hours in the gym, Johnny. They don't look different.
Oh, there's another spider rad right in front of us. Did you see it over here? Yeah, right there. I can't believe how many there are. Is there a way to get that in focus or zoom in on it or something? I think again it might be two of them together. That's really neat. It's like we always see two together.
Red blood bugs.
Oh wow. Ah! They're so beautiful. if we can collect that, that would be nice.
Section sampler, or do you wanna?
Yeah, whichever's easier. I think they'll survive of the section sampler probably, but if you want to try the D, that would be
I'll set up
fine
a section
too.
sampler to the side of it, so that we can do like whatever.
I don't know how
Exempt itself.
strongly the section sampler sucks. Like if it's really, really
Good.
strong, then it probably destroys them. But
It looked, from what I saw, what I saw went in, it looked like it was intact.
okay. Well, then if it goes in, and once you see it, if you could just turn off the suction immediately, then it won't smush them up against the top. Let's see. Go in. Ooh.
Oh shit.
It wants to go in the D.
Yeah.
Is it in there?
It looked like it was in there, so we have to.
All I did was shut it while Chris was trying to do the arm.
Yeah.
Yeah, but it looks like it from the view that we can
Can
have
we-
pull back and see.
Is there a way to see the look inside of that one?
Yeah.
Try and zoom in on it, Chris, and see if you can see it.
I don't
There's
know.
no way to really tell if it's hiding, if it's in there or not.
Yeah.
I don't know.
Swim or fly
If
around
you
a little
look-
bit, see if it.
See it.
Yeah, I'm not sure. Let's see it. Well, I'll mark
Got an
it.
idea.
Which one was that?
6. What are you trying to do?
Wanna let go of this?
Okay.
And look at it with the camera on the jaw.
Okay.
I was gonna try to put it around here, but
I can extend that if you wanted to.
probably.
I don't know if we got it. I guess we'll find out when we.
I'll have a way to try to peek in there a little bit.
Oh, I do see it. Actually, do
Yeah.
you see it? It is in there. Oh, look how small it is. That's
Where?
crazy.
Oh yeah, look
Oh
at it.
my
It's right in the
gosh!
middle.
Whoa!
Nice catch.
Yeah,
Nice catch.
nice. Oh, that's so cool. It's so small.
Give him credit.
Wow.
All right. You can still that arm.
It
out.
definitely wanted to be captured.
Did it though?
Oh,
Fitting for
yeah,
Valentine's Day tomorrow.
yeah.
That is all the way retracted on that.
That's fine.
Start with that. Okay, cool.
I'm sorry, I had on the wrist cam. You guys were zooming in. Can we get one more zoom?
It was It floated out
Yeah,
of
it was
view.
floating in the-
Yeah, it was
yeah.
floating. That's neat.
Is that it?
No, I think
No.
it's behind the black thing.
It's hiding.
Yes.
Okay. Mm.
Come to your starboard, please.
I'm doing that now.
Oh, do you see this black? Is that just on the screen, or is there something actually black in front of us?
Where?
Well, right. Oh, yeah, there's definitely there. Something's right there, right? something. I thought I saw. Oh wow, there's so many of them. Those spider rads.
Yeah.
So cool. I thought I saw a black thing, but I think it's gone. Or like it wasn't. But if we can get more spider rads in the suction sampler, then I would definitely be excited about that. Do you see it?
I do not.
Yeah, I don't. I think it's.
But maybe we'll
To
find
the-
it. If we, as long as we get it out and ready, maybe we'll find it out.
Oh yeah. Okay. I can't believe how many there are at this depth. So many.
And then we'll start again. Alright, let's see if we see more of those. Lots of those Salps.
Those radiolarians can actually eat copepods, which is really cool. I mean, they're protists, but they're eating
But
animals.
There's a lot of stuff right in that picture.
Yeah. Wow.
That's cool.
This is quite the depth. Oh, there's one to the left.
Where?
To the left. Oh, right in front of us. Oh, look, and there's another couple one right in front of us. See the two? That might be easier to get. These two right here. Wow, I can't believe there are so many. It's wild.
Hmm? Oh,
Inside.
wow, okay.
Mm.
It's coming out toward you, Chris. Like right about there is outside the jars.
Oh, there it
There
goes.
it goes.
I'm watching. Oh, there they are. I think they're in.
Yeah.
No?
Flip the one in.
Something went in.
Well, I don't know.
Let's see.
Oh, I guess there's one that's still out. I don't know.
I think that was a third of our ranger.
Oh, yeah, they're in now. They're in now. Oh,
Right.
they got
Yeah.
sucked out.
No, they can't get sucked out.
Oh, well,
It's
they're.
one way in.
Yeah, they went in
It's up
and
against
then
the,
they moved really fast somewhere.
It's up against the
I think I said,
filter.
yeah.
Okay. Well, we might want to rotate the jar then.
Okay. That is jar 2.
Oh, I gotta mark that, huh? Biology. That was section sampler 2. Wow.
Send a picture.
Oh my gosh. Wow, that's so cool.
You want those as well?
Huh?
Last one,
Would you say?
Did you want those? You want those as well?
Sure. Let's just, take them all. We'll just empty the ocean of them. Wow, I can't believe how many. It's so cool. I remember the first time
Well,
I
we
saw
do
this.
need it.
Uh-oh.
Not even close. We're on the jar. I know exactly where they
They're in
are. Alright,
front
we're
now.
coming out now. It's too close to us, Bruce. It's all the way by jar 34. Just hang tight. They're really hard to see when they're far away. It's coming across the top of jar 5. See that right out
Yeah.
front?
There it
Got
goes.
it.
Let's see it. Wash into the jar. I saw it. I think that's
Stop.
it.
Stop. That is off. So
Which jar was that?
it's swirling
Three.
around in there.
Yeah, I
Three.
saw it.
Is it 3?
I'm gonna index. Okay, indexed. Alright. Good there.
Alright. Alright, 52.4. Moving into the northeast.
Yeah.
Hmm. I think the genus of those Fodarians, they're not actually radiolarians, they're actually Fodarians, and the genus is definitely known. I do not know the genus off the top of my head, but I can find that out. There's so many of them. It's really cool.
Need to check those. Come back up. I don't know if it's just 'cause where they're at. They're a little shaky.
Oh, 5 and 6.
May just be whoever's catching the water.
I just texted my colleague Natalia Lopez Monferrer, who's an expert in Fodarians, and we'll see if she tells me what the genus name of those Fodarians are. Wow, that's cool.
Is Tony calling you?
Go ahead, Rich.
Hey mate, I was just wondering if there's a reason that the water is on at the talking head.
Nothing that I.
Nice, and as we're covering.
Yeah. Yeah. Okay. Could we keep coming up? So we're just rinsing the salt water off as we do.
Okay, we'll do that.
All right.
If they want it off, we'll turn it off. I was looking at that.
Something about that, Jim? Yeah? Yeah. Okay, 2 people. Okay. 2 people.
Something else.
What are you putting there?
There's like 7 days in right there.
Oh.
Make sure we're there. Okay. We're moving them really close. I keep moving them north. We're really trying to dump them so close. You alright?
Yeah, okay.
We've been collecting a couple things in jars as well as in jars. So over there. So let's do that. Alright.
I don't see
No,
anything moving in there.
nothing moving.
Oh, there's one. There's one swimming in there. See that? Cool. Wow. You can see its fecal pellets inside of its body. Those like glowing white things inside of the like what looks like the head,
Oh,
you know?
yeah.
That's its fecal pellets.
Oh, wow.
Yeah. And they're really distinctive shaped. They kind of look like footballs.
Cool.
So when we see them in the sediment traps, like the larvation fecal pellets are just like really obvious. And there's always a lot of larvation fecal pellets in,
Oh, wow.
in the sinking stuff.
Colleen,
Yeah.
do you want dinner?
Sure, yeah. Cool, thank you.
Mm-hmm.
We got lots of
wait,
Fodarians.
Yeah, what do you? Oh yeah,
Tons. Or like there's
Nice.
so many of these really
the
cool.
same kind. Or?
No, it's like a different one. They like Steve calls them spider rads. They're like have these appendages. Maybe you'll see more. Anyway, they're really cool.
Cool. Anything else in particular?
No. Yeah, see there's one right there. See it?
Uh huh.
Left.
Spider rats, you said?
Well, rad.
Rad. That makes
But
more
it's
sense.
a Fodarian. Spider rat. You
Oh, I
know
saw
a
this
spider rat.
earlier and assumed it was a
it's
jelly.
so beautiful. No. It's a Fodarian.
Wow. You said we already got a couple of these?
Yeah. Well, we have so many collection jars, right?
Why not?
We should, if we can collect them, let's collect them.
Awesome.
Because, we can sequence,
Oh.
them. We'll see what they're eating.
Mm-hmm.
Okay, I'll go eat.
Okay.
So just keep an eye on the vessel because you're definitely drifting towards it. So you gotta keep it moving.
Okay, 2 on the left.
Yeah.
Great, new waypoint.
Copy, new waypoint.
What speed you want him to go?
Sorry?
What speed you want him to go?
Very little. Yeah. Sorry, I'm just filling in. I don't know what they have been doing.
Same thing like what we were doing, slow.
Okay, yeah. Because we still have a bit more time and we want to stay.
We don't want to come up at 8, so
Yeah. So we don't want to come up too fast.
we don't want to come up too fast.
It's almost
So you don't know, you could go forward.
Can we
come
look at
back.
this thing in the distance over there? It's like a Siphonophore, I think.
where? Point from it?
This guy. Do you see it? Yeah, the chain.
Go on.
Oh,
Right.
Nope. I think we switched it.
Let's see what else.
I think the Baltic turning off-road also.
Let's look at this thing. Oh, another larvation. Nice.
Want to keep looking at that?
Well,
It's up to you.
He made the decision for me.
Maybe not.
They're so delicate. There's a fish over there we might be able to look at. Flashing thing. Oh, never mind. It's a arrow worm. We don't need to look at it.
Nope.
Yeah.
Okay.
Look at the
I mean, I don't know what they look like. Wow, I did not even see we're there. Like, if it's a fish, they look different from each other. All keognaths kind of look the same.
I got a red one today,
Oh,
but it was.
never mind. I stand corrected.
Real large. It looks like. So I
If
don't
we
know.
see a red one, I want to zoom on it. Hey, did you guys? When did you get a lot of this trawl?
It's pretty sparse because we're in a pretty low nutrient area.
Yeah.
We get a lot of cyclothony, big boys.
Shockers.
Pretty big ones. A lot of hatchet fishes, which we have not been.
Saw some on the way down. So that tracks.
We just saw a heteropod sea elephant in our movie, which we also
Nice.
got a lot of.
I love those. I think they're so funny.
There's like one guide from Australia, the Tasman Sea, and they call them jellyphants. Like
I love
sea
that.
jelly elephants.
That's great. Have we zoomed on any fish or anything?
There was a cyclothony while we were processing. That's all I really saw.
Oh, let's look at this. Four.
Huh?
Yeah, no, we can keep looking at it.
Oh.
Well, I think it's up.
Yeah.
There you go. Oh, I guess it's pretty small. Okay, it's gone. Yeah, let's look at that. It's probably a fish, so it'll probably swim away. Maybe not. Definitely a fish.
Cyclothony.
Is it?
Yeah.
Oh. Yeah, Cenozoic. You're right.
We got close enough to ID.
The most abundant vertebrate on the planet.
Mhm. What's their depth range? Like 1,500-ish?
It depends on which species. So
Okay.
you have shallower species, and then also deeper can be below 1,000.
Okay. But do you ever really see them in surface water?
Like the upper 500 meters.
Yeah.
Yeah. It's funny, there were so many salps, maybe 100 meters deeper.
Oh, really?
And now there are no salps.
Did you get many in the nets?
No, which is very strange because they were all like everywhere.
Huh. Can we look at this thing? I think it's kind of small, but if we can. Well, then maybe we should. It'd be cool to collect a salp then. But except that now we're not seeing them.
They were like whole chains.
Oh, cool.
Huge.
Oh, it's a Foram. Cool. It's just a small one. So this little
Was it
pom-pom
really?
thing is like a single-celled organism that's just really big for being single-celled. What are you gonna say?
Radiolarian or Foram?
I think these are, well, okay. That one looks like,
This thing we collected in the trawl and I called unidentified flat blobby, and by the time Alejandro had time to take a photo, it had already melted into
yeah, that one I think was a radiolarian. The like, kind of like fireworks ones,
Uh-huh.
I think are Forams. But
Cyclothony.
yeah, they're all Rhizaria, so we can just say Rhizaria. And they're all like single-celled protists that get really big. Colleen said they collected a few.
Yes.
I'm surprised you got them in the net at all without it like being totally wrecked.
The flat jelly thing.
Yeah.
Yeah, we collected a bunch of Collidaria for
Mhm.
Colleen. Well, not for, but there were a bunch of them in the net.
Yeah.
They're
Nice.
pretty intact.
That's cool. Yeah, so Phaeodaria are actually just a type of radiolarian. The taxonomy for protists is like messy. But it is a type of radiolarian. And a lot of the larger radiolarians we've been seeing are Phaeodarians. Ooh, let's look at this thing. I think it's just a discarded larvation house. Maybe. No.
Is this one of these Fodarians?
I don't know, or it could be like an egg mass of something. But that's my two thoughts.
The ones we collected today, the ones from
If we
the
could get a
last
zoom on that, it would be
trial?
great. I know it's really small. But
the last trawl, they were like jelly bean shaped, and this
Mhm.
trawl, they were really long and flat. So
Huh.
I don't know why they are different shapes or lengths, or if it's a different type.
probably, yeah, different species. Okay.
Your mom's here.
Hi, Mom. That's another Foram.
So, focus.
October. We are almost 500.
Oh, 5 minutes.
looks like the focus got a little bit off. Yeah, that's better, thanks. Can we look at these two things that are going by right now? If we can stop times, thanks.
That's definitely great.
Mm. I think it's to the right still. They might have become unattached. Hmm. Yeah, I think they're gone. Whoa. That's a little jelly. Little
Was
jellyfish.
on the rush.
Yeah. Did not want to be seen.
More than that. What came from this?
They can be surprisingly fast, the jellyfish.
What they want.
Yeah. Yeah, it's a little shrimp. Just not gonna let us look at it. No. I mean, you can see how big their antennae are. They're like
Yeah.
very sensitive to movement.
It's like the blind fishes on the cave.
Mm-hmm.
Anything you move nearby, however, you try to swim very gentle or use the bottom.
Mm-hmm.
If you
That's another larvation.
what's that called?
No.
They were the same one.
Yeah. They're really tricky. I feel like unless they're really big, it's hard to get close enough. Jellyfish in the distance. Or maybe it's a amphipod actually, or a shrimp. Yeah, it's an amphipod. Because we're big and loud and bright. It seemed like a predator. Yeah. I feel like on the ship we don't notice how loud it is because the ship is so loud on its own. Mm-hmm. Oh yeah. It's still so loud. So there have been some technologies developed to sort of drive the ROV automatically, and including following things, but a lot of the time it's just not as good as what the human pilots can do. For example, when we were doing the transects earlier, like they have like a auto depth setting that they can put on, but they were telling me that like
All of that does that.
yeah, it moves up and down more than just like a human driving
The
it.
only way for you to the auto that to work perfectly, the vehicle must be totally neutral.
Mm-hmm.
Because if it's heavy, every time it falls a bit, hit.
Yeah.
If it's positive, it's the opposite.
Mm-hmm.
Can be the most advanced one, we will do that.
Mm-hmm.
Can be the Bentley of the oldest. Like the Chile, we call the Bentley of the oldest. So, what you can do.
Oh, let's look at this radiolarian right here. No, they just go above and then they disappear.
catch up with the rest.
Okay. Is the ship just staying in one place? Or is it moving?
I think she's drift because the station is here. The ship is out here.
Mm-hmm. I don't know if that was.
Yeah, it was brighter than the other ones. That's why I pushed towards.
Yeah.
But the same thing was moving.
It might have just been like a really small fish. Let's look at this jelly down there. Or not a jelly. Some kind of protist, I think. That looks like maybe a very old. Well, actually no, I can't tell what this stuff is. Oh, it's like a really big aggregation of.
No, it was.
Well, of like radiolarian material maybe.
Same than the other one. this one?
I think so. It's hard to tell. Yeah. Ooh, let's look at this. I think that's another ctenophore. Yeah.
That's it.
This looks good, yeah. Oh, yeah. It's not as bad as it was the other day.
Yeah, I got worse. So
Mm-hmm. Oh, tiny jelly. Yeah. That's a big siphonophore. We can keep going.
keep going.
Yeah.
We saw some big ones.
Mhm.
Nice before. Very nice. So that push
On the way down, we saw a really long one.
Push a little bit forward because the bus.
Mm-hmm.
If you see something, try step on the brake.
Okay. We can try to look at that fish. Looks, or that one.
That one.
We can also keep going if we need to keep moving. Just looks like a decent size. Maybe it's an arrowworm. Yeah, they keep fooling me. It's not a fish. We've got this thing on the lower right.
Mm-hmm.
Yeah.
This one?
Yeah. We try to stay farther from stuff and zoom, because I feel like
Okay.
we get close and then it just,
Strong.
yeah.
There's a current to the right.
Oh, that's true.
I'm trying to be closer because the zoom.
Yeah.
kills me.
I think that's like an.
Sinking house.
I think so, but it looked like it had some of those like glittery bits, like the forams. But
Forams.
maybe it had gotten caught on it. Just like a cool piece of detritus. Sorry, all pieces of detritus are cool.
Some are just cooler than others.
Have you seen any I know you're looking for bioluminescence on the sediment trap, is that right?
Mhm.
Have you been able to see any of that?
Yeah, I got the most at the first station, where I actually got some like detrital bits that were glowing. The second station, most of the stuff was like copepod carcasses or amphipod carcasses, which is also interesting, but it's like a little bit different. Because things with chitin, they will often grow bioluminescent bacteria after their death. They really seem to like that, so that was really cool to see. But also the Fodarians that we've been seeing, which is a type of those big single-celled radiolarians. We got one from the ROV, and that one was still glowing.
Nice.
And then we also get like little bits of them in the gels. But we'll see at this station because this is going to be even less productivity than the last one.
It's totally different when the water gets more bluish and less nutrients.
Mm-hmm. It's cool that you can like literally see the difference in the color. Oh, there's another. Can we try these two? Might have the same issue, but it's just really interesting to me that we seem to find them in pairs. This type of radiolarian.
They kind of like stick figures.
Yeah. I think those are the spider rads.
Yeah.
But like I feel like I've seen a number of them where there's like two of them, and I wonder if it's like a coincidence they just get stuck, or if they like it's one that like reproduced and split apart, you know. But I'm just making things up, so I don't
How
know.
do they reproduce? Is it asexual cloning?
Probably. I don't know. What?
They're protists, so they're gonna divide.
Yeah.
A magical.
I don't think they're radiolarians. Just for the record here, they're doing
What do you think they are?
Fodarians.
Fodarians are radiolarians. What? That's what
Not anymore.
worms
anyway.
told me.
Not anymore.
This is why I said protist taxonomy is a mess. Like they keep changing things around. Okay. I'm wrong
Is
what I
that
said earlier.
What are these? Oh no, this is just like
I think it's like a piece of a Siphonophore. But they are Rhizaria, right? Fodarians. Okay.
I think it's called Circozoa, where they now go.
Yeah. Oh.
You have the technology.
It's another larvation with one that's still inside of it, swimming. You can see the tail beating.
Some from the first ones. Okay, 2 hours.
Yeah. Oh, the taxonomy changed in 2004. I'm way behind the times. This isn't even a recent thing.
Circozoa, which is the other group of this area.
I
That's
don't
where
like that
the
name.
Fodarians
It's like circle animal. That's a. I don't think that's a very good name.
Well, not circle with an E.
Oh, circle. Okay. Well, they're all Rhizaria.
Well, there. We got a smart guy. Says
They're all
changed in
critical.
2004.
Mm-hmm.
Well, there you go.
I don't even have an
That's
excuse
not
of it
that
being a
recent,
recent
no.
change. I don't remember where I got that information, but it was wrong.
Okay. I can relieve you. Sorry, that was a long dinner.
It's okay.
Thanks for giving me a break.
Oh yeah. Well, and those like Fodarians
Mm-hmm.
that are like the spiders.
Yeah.
I saw a few of them where there are like 2 of them.
Yeah,
I
they
think they
seem-
like recently divided.
I mean, that seems pretty logical to me. But yeah, we keep seeing them as pairs.
Yeah.
And in Monterey, we see them that way too,
Okay.
as pairs.
Did
So
you collect any of those?
yeah.
Oh, okay, cool. Because I saw you had written forums on there.
I wrote forums.
Oh. Have you
I
have you
collected
recorded
2 forums.
somewhere? what
I
we put
logged.
in one?
I didn't write it down. I put it in the log, the
Okay.
this log.
We just want to get it before we get the
Okay.
RV back. I can try to.
Yeah, I can
Track it
just.
down.
I can. It's I can look at it in the log. Oh, let's see. I think. Oh. Okay. So I don't know if the person who asked this question is still watching this video, but someone asked what the genus of those Fodarians is, and my expert colleague Natalia Yopes Monfrere thinks it could possibly be this one called Collidarians, which is like COELODNDRIDAE. Possibly. So, if you're curious. It's not much known about those. So the log.
You guys are on track to be at the surface around deck 8. Yeah. It's an hour, right? Hour, 10 minutes. Right.
What's the next step for the
The next what? No, it's a zooplankton. We do have a few zooplankton appetites.
The 8 come back, and then we speed up. The problem is, but the problem is if I speed up,
I know.
Look, that is just the current.
I know. I hear ya.
Speed, I even turn my head for the cars to don't come.
Yeah, I know what you mean.
Because if you're facing current, look at the speed.
Yeah, I know. Well, yeah, gets much higher.
Yeah.
Yeah.
I was trying different headings to try slow down the movement of the water.
Yeah, I know. As you get closer to the surface, the current speeds up. That's what happened before. We got to 400 and we couldn't do any more collections.
Because it's too fast when you see.
Yeah. So you just have to make sure to be heading up by the time. You know, you can do whatever you want until that point.
It's been written down, but I can get it to you. What are you looking at? What are you looking for? Yeah.
That's where I wrote down my collections.
Okay.
The Forams, and then I lost those, of course.
Okay. Of course. I forgot
Those are my
to.
collections of the Forams.
Okay.
3 and 4.
Okay.
And those are for Josh.
Yeah. Well, I mean, I collected them. But
There's one that I forgot to log.
Oh,
Sorry.
that's why I
Well,
didn't
we'll
get the
we'll see it when
PIs
we
during
open the
this.
bottle. When we open the jars, it'll be like a little surprise.
What did you collect?
I think I feel like there's a missing.
I mean,
There's definitely a missing
if you're a
rad.
little late, it's okay. But
And a missing
Doug has
peanut
someone
butter.
to do it. That's why we're on deck at 8.
Oh, okay.
Because of Doug, right?
Okay, cool. Yeah.
That's why it's 8 o'clock.
What?
I don't want to keep him
400 meters.
barking.
You never saw the joke of the door?
No.
They showed the door.
Oh, okay. Okay. I'll look.
Yeah, so my plan is just to like see where we can.
It's a freak to them.
Okay, I'll have a look.
What are these Ketignaths?
Yes.
There were a lot of kitty mags.
Oh, cool.
So what to do?
So how much time do we have?
Now we're in 5 minutes, 10 minutes.
Like before we need to like actually come up?
On the deck.
Do you have any depth preference?
Yeah, it's deeper, like deeper than right.
Or you
Or
prefer do slowly now and then I speed up because we have few
Yeah.
minutes?
yeah, I would prefer to stay at this depth.
Okay.
Yeah.
I'm gonna move it very slowly.
Yeah.
There's a lot of things kind of hanging out around here, I guess. There's also stuff up there.
Let's just kind of like go straight and like see what's here.
Go towards the vessel.
Okay.
How do you want to stay at this depth, Colleen?
Just so we kind of can look around, just like so we're not just always moving up. Like
You just get to this depth?
I think he was just slowly moving up.
Yeah, okay.
I'm
So-
believing that the vehicle floats very,
Yeah.
very, very slowly, not even
That great.
0.1, 0.2.
I was only
Sounds
asking because
great.
I'm curious if you have something you're specifically looking for.
I mean, I think it's easier to like see, like yeah, I'm looking for the rads and the
Yeah.
and the forams.
And you think they're better down here?
Yeah.
Or it's just easier to
Yes,
collect
I think
down lower?
they're more down here.
They're easier to collect
Yes,
with lower current.
and they're easier to
Yeah.
collect. They're usually at this depth and deeper, and they're not going to be as like the
Yeah.
Fodarians aren't the big Fodarians aren't usually in the shallow depths.
Right. Yeah, for a while I had them just like very slowly creeping up as they went forward just so that we would make some upward progress.
Yeah, but then at the end he can just go
Yeah,
up.
yeah. No, I'm not saying you should do that. I'm just telling you that's partly why they were doing that, I think. You know?
Yeah.
Did you guys slurp anything?
Yes, we did. So we slurped some Fodarians, so we'll see
Oh.
how they survive.
Okay.
Yeah.
Do they all go into the same jar?
No.
Okay. Just sometimes. Rachel, you
Catch
gonna
and
be
chew on
able
the
to
same
look
jar, we must have
Same
very
one.
lucky.
Yeah.
ROV, or do you need to like go do something else? You can go do something else.
You need help?
I think we probably have
No, it's
to
great to have you here.
Oh,
No, I was just asking if she can help with sample collection. from
Oh,
ROV,
yeah.
Because it's actually kind of a lot.
Oh, yeah.
Sure. But if you need to rest or something, you should do that.
Times of Marcus, so yeah,
I mean, if we can use your ladles, we can do it.
4. Okay.
Yeah.
I think we can
Well, let me
ladle.
know. I think I'll be around. So ladles are probably in the folder.
Can we see what this is?
Okay. And also those like smaller.
Right there.
Yeah.
Because it's you'll do the jelly scooping.
As long
Wait,
as
oh,
I
it's
know
probably
where
just a
those
little
spots are.
mucus
Yeah.
thread, huh? It's so small. Yeah. Cool. I still think that's cool. You can keep going, yeah. I just, like some small things you kind of have to like spot far away, like this, like it's fine, we missed it, but like the small things that are cool, like that Foram.
Foram, yeah.
Yeah.
Josh would want that.
Yeah, so, but like, I'll just keep looking. I'll spot it when it's far away.
Colleen, we saw something and I didn't know what it was earlier. We didn't get a super good look at it, but it was like that like kind of net thing that I was like, is it an egg mass? Is it a radiolarian? Do you remember that?
Oh yeah.
How would you describe that?
It had I think it was broken. There was a hole in it.
Yeah, there was like
But it had a
Oh.
bunch of little
it was
dots.
kind of like a pancake shaped net.
Huh.
Not very helpful.
But it wasn't a ring?
No.
Yeah,
it was
okay.
Flat.
Huh.
Are there Fodarians that look like that?
Yes. That are pancake shaped?
There's a farm. Oh no, it's not. Never mind.
Yeah, that was it. Yes, there are some flat ones,
Okay.
but I don't know if that's what you saw.
Like the Collidarians have that like the little dots inside of it.
Mhm.
Kind of look like. I mean, you were closer, so maybe you got a better look.
I mean, it looked like some sort of rosaria to me, but we've established I'm really bad at protists. So.
Let me see.
I was like spreading misinformation earlier by accident.
Outdated information.
Yes.
Oh,
Information that's been wrong since 2004.
what were you saying that was
I
wrong?
was saying that Fodarians were a type of radiolarian.
Oh, well, it's confusing. And plus some people still think they should be called radiolarians because that's what they were originally called before radiolarians. I don't know.
It's like when they
Yeah,
changed
it's
the name
people.
of
It's
Crenarchaeota,
fine. Doesn't matter.
Some of them
Oh.
to Thalmarchaeota.
That bothers me.
It's a nightmare.
The most important thing is that you understand their evolutionary relationships.
Names are just names.
Yes, it's
Years.
fine. It's like as long as we know what we're talking about and how they're, how they evolved relative to each other.
Somewhere around here, if it's possible.
There's definitely less at this depth than when we were down at 600 or so. Yeah.
Could it be that it's because the migration?
Maybe, yeah. Well, no, we were seeing like Siphonophores and,
Oh.
well, maybe the Salps.
Then we need to push forward a bit.
Okay.
Then around 100 meters, maybe that's.
Okay. Yeah. Let me know when you just have to catch up to the ship now.
Not too much. If you need me to stop
Okay.
and I can step at the braking time, we can try.
Okay.
Because as
I'll
I
let
go
you know.
faster forward is more different. But
Yeah. Okay. If I know I gotta be eagle eyes. I might tell you to stop when it's not interesting.
don't cost any extra money.
Oh, okay.
Same price for the ride. The rest is-
Yeah, that's actually. So somebody when we were here was asking why that would be the case, and I was postulating, but I'm not sure I had good answers because it's not really my expertise. But I don't know, something maybe they'd be interested in hearing about.
I think it's usually has to do with like occupying specific ecological niches, so there's less entries in the pool. If you all have the same strategy, it's hard to compete with each other, so they start to like diversify. So it's like feeding strategy, and then there's something about that drive to evolution and some of the other species that you kind of have at different parts of the water column where feed tend to be more active in the histories than above.
That's an place with deeper nutrients. Yeah.
So like if you think about California current, it's high biomass because a lot of this, there are few groups that represent a large portion of the community.
Yeah. I mean that's how I think about it from the point of view of phytoplankton, and a lot of times when there's a lot of nutrients, it's like there's something specific, like one thing, and then the food chain that follows from that is much also much more specific, and there's plenty of it for them, and so in equatorial places, it's like more of a mix of things growing, maybe nothing, so everybody kind of has to figure out the way to.
Maybe if you, I guess it looks like from the echo sounder that it's at, or EK80, it's like, am I interpreting this correctly, that it's at like 250 there's a layer of stuff, so maybe if we just come up to 250 and hang out there for the rest of the time.
Yeah, because
Okay.
some do the same thing for like why do somebody has a fish? It's like why do somebody has a protein, but phytoplankton.
Because I have a short attention span.
Well, identifying crustacean is like one individual, one species.
How did you do the transects?
Yeah, well, you know, just start chit-chatting.
Talking about poop.
Yeah, yeah.
It's coming to 250.
It's amazing.
250.
It's coming up to 250.
Very cheaply compared to Siphonophores.
You what?
The nice things. Pops up
Mhm.
when you're speeding.
Yeah, and there's no way to stop for them.
Looking 240 or 250?
250.
250.
I mean, yeah. I just figured we would spend the time we have left just driving straight at 250.
Yeah. I was looking at the migration.
Yeah,
I mean, it's still going, but
because we don't have that much time, so might as well just go where seems like the most animals are. Okay. Well, yeah. Okay. Yeah, we can just, I mean, this seems like a pretty good speed to see things before they run into us. Yeah, okay. That's just the current. Yeah. Okay. Yeah, because actually there's nothing at 256 meters. It's all at 250. Yeah.
But you ask me, but you know, autism doesn't allow this. If you ask me 250, doesn't allow me 250.
Yeah. Okay. Oh yeah. Too close to stop. Can we see what this is? Right there. Yeah. Whoa, what is that?
Oh, we saw some of this earlier.
What?
Is it like aggregation of
What?
foreign bits?
Oh, huh.
I don't
You know, Alex showed me a video, like, if we could suction sample that, that would be cool.
Yeah.
Can we try? I wonder, Alex showed me this video from a previous cruise where they had, he said that they were radiolarians, and I've never seen that before.
'Cause it's like, are they
Yeah, let's-
dead or are they doing that on purpose?
I don't know. Let's suction it and then we can look at it under the microscope. If possible.
I think it comes apart pretty easily.
Yeah, but still, its bits will be in there. We can still put it under the
Some of
microscope.
those needles will be.
Yeah.
It's pretty big.
Suction.
Yeah.
What are gonna use?
All right. And then as soon as it gets into the 4, as soon as we see it go in, I think you should try to turn off the suction so that it doesn't smash.
It's gonna staining hose.
Ooh, it's breaking apart. Might not get it.
Oh, I can still get some of the bits.
Maybe. I'm just really curious if that was radiolarians, if that was the same thing that Alex showed me a picture of today. Because it's so, see how the way the light is kind of like reflecting off,
Like
or
glary.
refracting? It makes me think it's silica,
But
right?
that's what I thought about those forams and there's
Oh, yeah,
Or
that,
try
and
to
that's
get
not
it.
silica.
You want try?
Yeah, sure. Yeah. If there's a chunk that's worth going for. Because it would be really neat to put it under the microscope.
Can you turn on the suction for me? So that we're gonna use it.
No, just turn it on and leave it on.
Yeah.
Which one you find off of?
Huh?
See if you can find it in this camera. like last year.
Someone in the chat knows exactly which dive the past dive, the Radiolarians were seen in.
Whoa.
Yeah.
Write that down.
Yeah.
He's put timestamp it out.
I know. Whoa. Timestamp. Jeez. Service. 640. Part 1, part 2. 2341.
There's definitely some fish here.
Yeah, I think it's gone. I think it might be gone, so we can just keep searching.
Okay, that's a no.
Can we see what that is? This thing? Boom. That guy. Ooh, I think there's another. We got another chance, right? Whoa.
Just turn it on slow.
Okay, yeah. Let's try to get that, because it would be. I'm so curious.
So,
No, it's not.
just turn it on slow.
We're gonna lose it.
I think that's Krill, right?
Is it krill? I was
See this?
told
And it just feeds a quiet.
suggested shrimps.
We just leave it on slow.
What's it a suggestion?
It's not sucking.
Suction on. Suction on, right?
Otherwise we could try it in the jelly jar. But
It's more, it's good.
yeah, I don't know.
Okay, right in front. Suction on.
Oh.
It should be.
It's on.
It's not sucking.
How can you
Sucking.
how can you tell?
It doesn't look like the little particles are
It's
going
not
in.
on. You don't see anything particles in there.
Hmm.
You turn it on. No, that's suction here.
Yeah, now it's going. It's going now.
What was it?
It went off to the like starboard side.
Too strong. I'm gonna make pieces.
There's a piece up here. Up there.
Excuse me?
I think that's one. Yeah, I think that's part of it.
Okay, let me see. See if I can see that. Yes, it's a part.
There. Oh, I thought it was going in. Yeah. Okay, well, I think we have to keep searching, and then maybe we'll find another one. What about this? What's that?
Same thing.
Yeah, let's try. Third time? Third time's a charm? Is it?
Yeah.
Is
so.
it is?
Looks like it.
Okay. It's not like a. Oh yeah, totally. Oh, too close.
It's actually you're inside of it.
Yeah, it's. Yeah.
Yeah, come back. Yeah. So if you put on this camera a little, you're gonna have a better gauge where it is. So you see where it is? It's right there.
You see it?
See it's going in the corner there.
It's down.
Coming on finally.
Okay, I see it. It's going around the front of the. Yeah, now it's on top, like in front, above.
Have you seen
Yeah,
that?
see that stringy thing? That's it. But I think it's low. It's below the ROV. There. Now it's coming up. Nope, that's not it. That's it. Right, really close to the camera. It's like very inboard. It's okay. I don't think we're gonna get this one. It's probably kind of broken apart.
I think so. That's
Maybe if we see another one, we can just zoom on it and try to get a really good zoom. I think that's a larvation.
Ready, ROV?
Yeah, go ahead.
Yeah, Tony, I just want to give you guys a heads up. We're going to start recovering here in about 5 minutes, and we're pretty close to surface, so I don't know how long of a time you got to give you guys a heads
That's
up.
the one.
Oh, let's see what that is. It's. I don't think it's another one, but let's look at it.
Roger.
Can we just stay far away and zoom at it, or?
I know where that might go. Do all the for you, no problem.
She wants to look at this thing here.
Yeah, I know. It's inside the porch, though.
Hmm. done. What about this thing right there? Maybe we can just try to zoom in on it, like keep it far away and then try to zoom and see if we can. Oh yeah, but look, there's tons of them here. Try to figure out what they are.
You want to go on that, please?
No. Say go E flat up. We'll do it.
Is there a way to change the focus or like control the focus or?
Yeah. You want to do it, Mike?
Yeah, please.
Okay. Huh. What? Yeah. Let's try to figure out what this is.
Same thing as the
Yeah,
Foram.
yeah.
Those look like fine.
I mean, there's a few things that come to my mind, but I don't think I've ever seen anything like that. I mean, the other thing I think of is like the like those diatom mats, you know, but it's so shiny. I don't know.
Silica is what would come to my
Yeah,
mind.
it's-
Except
it could
that
either-
those forums were also shiny.
Yeah, but I don't think it's forams, is it? No, because that's like more of a radial pattern. This is like
But do you
a
think that- I guess-
mesh. This is like a
I think that's one live
stack of-
thing, or
No,
like
I
bunch
think
of
it's a
dead
whole-
things aggregated together.
I don't necessarily think it's dead.
Oh, but like a bunch of
Like there are these diatoms, like rhizosolenia, that grow that like form mats on purpose,
Mhm.
and then they use that to move up and down.
yeah. So it could be that.
Maybe, but I had never seen that radiolarian structure before, so it,
Maybe
maybe
on the next dive we'll be able to get some.
maybe. Are you done? Are you have to recover now, or can we try one more time to collect?
You got 4 minutes.
Okay, I saw one. Okay, let's try one more time to collect. How about that one right there? There's so many of them in the water. It's this thing right here.
You want to dissection sample?
Yeah, whatever you want to do. Maybe whatever you think you can do better. If you can get it in the jelly jar, then that would be even better. Oh. Uh-oh.
Is you more on view?
See one?
There were a ton around here. This one, right there. Coming up pretty fast. All right, it's inside. It's, yeah. Can we back up or?
It's pretty much it all the time.
Yeah. I think it's got smashed. Okay. Probably just have to approach them really, really slow.
Yeah, if we're gonna be on deck for 8, we're done.
Okay. They just will remain a mystery. Maybe we'll see them tomorrow or whenever we do. I don't know when we're doing the next dive.
Tomorrow night,
Tomorrow night?
but it might have changed.
I don't think that's. Is that one? I'm not sure that's. Yeah, it is. Do you see it's inboard? Do you see it's really close to the camera? It's not close to the suction sampler. It's over here now.
Having thrown that.
Yeah, I got you loud and clear.
Right.
Alrighty.
Wow. That was cool.
There's probably baking something out somewhere. Smells wonderful.
On that.
Are we recovering now or are we?
Yep.
Okay.
You're gonna track that rail. Bridge, that's the ROV beginning recovery.
Oh, go big, thanks. I'll get a guy for shot.
Thanks.
Wanna hand it over?
Yeah, give me a minute. We're still stowing stuff.
Okay. What's your depth?
It's only like 200, 220.
Yes, sir.
All right, Chris, you got command.
Roger. Alright.
Launch recovery mode on Mike.
Coming up.
When should they move?
Pretty much now, I would say. Don't keep coming up, let him come up some more. You want that boat with the flop over the back, okay? Have you done a recovery? Okay. So we want to come around, face opposite of your launch point.
Your umbilical is straight a bit down.
Roger.
Very odd. Very different thing than what's been happening for a long time.
Okay.
Yeah. Yeah. Fortunately here there's like no current. Or unfortunately, depending on how you're looking at it.
What?
I mean,
Sometimes help.
Oh, that's the
take no
thing.
current.
Yeah, that's totally different. Actually, I think I would register it. Which would be really cool. Yeah, it's totally different.
Just you concentrate on flying. I'm just making sure these are all closed because we've got problems with them coming open.
Nah.
Maybe this is the life.
I don't think so, because I didn't see the little circles.
Yeah.
It was all, I think it might have been a Rhizostolemia mats. That's cool. Yeah. I mean, most people have studied those through scuba diving, not through ROVs.
Oh, the camera.
Nice glasses you got there, Keith. Some nice glasses. Safety first. Okay. Yeah. No. Keep them on. Yeah.
You can come up faster if you want, Chris.
23.
Oh yeah, you are. Usually it's at 40.
Really?
Yeah,
Okay.
because that's meters per second.
Okay.
Whoa. Now you're kicking.
I forgot. Once this is on deck, you're off ship. Never mind. Understood. Got a little bit of slowage up here.
Swell, yeah.
What jars do we have on the suction sampler?
Looks like 1, 2, and 3.
And 4? No, just 3?
No,
Okay.
it's indexed to 4, but there doesn't not showing anything sucked
Alright.
into 4.
Both quads have had 4 figures, so just verify.
Oh, okay. Maybe they got something for it. I didn't see.
I don't know. Just 3? Okay. And it looks like we have
Floats on surface.
floats on surface.
Gotcha.